Good afternoon and welcome to Let's Crack NEET UG English, and we are almost completing this chapter, that is respiration in plants, and we have to just talk about few important things which are left, that is ATP gain, shuttle system, and then we will have a recap of aerobic and anaerobic respiration also. And in tomorrow's class we will again talk about just few things which remain, and then we will have a doubt solving session followed by a quiz. Okay, so students are joining in, and I know this is the time when the students finish one class and then they come to this class, and that's why there is an overlap of couple of minutes. Anish, hello. Rahul, hello. How are you? Hiren, good afternoon. So how is everyone? Okay, Nandini. Hello, Nandini. And as I said, it takes a minute or so for everyone to join in, because I guess they have the other class. Okay, Nandini, that's good. Oh, Hiren, you are having cold. Take care, because this is the time when the season changes, and this is the time when you're likely to catch infections. Ma'am, will this timing be changed or will it remain same after 31st? See, I'm going to share a few things with you when the class starts. I have a couple of important announcements to make, so at that time we'll talk about it. The timing and what is the next thing that we are starting. Okay, Hiren, take care. Okay, you've seen my channel, Anish, that's very good. And I hope all those things which are available are helping you in your preparation. You know, Hiren, you just now wrote that you have a bit of cold. Now this is a very common phrase, that I'm suffering from cough and cold. But as biology students, and you know, if somebody says that, you know, I have cold, everybody takes it casually, that, oh, it's cold, what is the big deal? But you know, if you change this word from the common terminology to the biological terminology, then you have to say that I'm suffering, or I have rhinitis. So when people hear that you have rhinitis, it sounds like, oh my God, what is this thing? Okay? So in medical terms or in biological term, the common cold, because there is inflammation of the nasal mucosa, itis word is used for any kind of inflammation, and rhino word is used for nose. So if there is inflammation in the nasal epithelium, we call it rhinitis. Right? So I'm sure you've heard of meningitis and conjunctivitis. No, Rahul, I'm doing that. Spelling is R-H-I-N-I, rhinitis, itis. Okay, Amisha has also joined. All right. So as I said, when we start the class, I'm going to tell you a few important things. One is that in the month of November, we will be, or rather I will be uploading lectures, or there will be live sessions only till 13th of November. And what I'm going to do is I'll record the lectures for you, and you may get it little later also, but after 13th we will not be having a live session. Okay, so this is one information which I wanted to give you. And up till now I was taking lectures on various channels like Biology 101, Let's Crack NEET UG, and this one, that is Let's Crack NEET UG English. But after rather 31st of this month, I will be taking lectures only on two platforms. One is YouTube, and that is this channel, that is Let's Crack NEET UG English, and the second is the app where I will be taking the Unacademy Learning app, where I'll be taking the special class. So there are only two places where for the month of November I will be taking classes. So this is one thing. Second is on 31st, I think it is Saturday. On Saturday at 2 o'clock, I have planned a special session for you, for all the students, and that is strategies to score 360 in biology. So that session is going to be at 2 on Saturday. So don't forget to join in, and I will be conducting that session on a YouTube channel, that is Biology 101. So I hope you all will join and will be able to take advantage of those things. Okay, Hiren is asking a question: in absence of air, life would be impossible, reducing anaerobic. Okay, Nandini says you'll be coming, Amisha will be coming. See, this is where I will be sharing my experience about students, my students who have cracked NEET with good scores, and what was that thing which they were doing, and what is that thing which you should be doing to get that good score. So this is what we will be talking about. We will not be studying anything. It is just a sharing thing, the informations or the knowledge that I have about the exam. All right, answering Hiren's question is, in absence of air, see, the life forms which exist on the earth right now, most of them are aerobic, but there are certain organisms which are anaerobic, and those anaerobic are obligate anaerobes. That means if oxygen is obligate aerobes like we. So we can survive only in presence of oxygen. No oxygen, will die. And there are anaerobes also. So anaerobes can survive in absence of oxygen. So depending upon which organism we are talking about, the answer would be impossible or slightly less. Okay, so let us get started with our topic, respiration in plants, and we will be talking about ATP gain, Shuttle system, and all those things. Now, I guess all of you are old students, so now you know my name. In case if there is anybody new, for them this was the slide. And the platform which we are using, I'm sure you're all aware of it. It is India's largest learning platform, Unacademy, and this is the app which I was talking about. So now onwards, whenever you want to attend a special class, you will have to use this app. You will have to install this app. This is available at your app store. And by using this app, you will be able to get live classes, quizzes, tests, etc. Now, see, I'm conducting quizzes on this app. And when we have a quiz on this app, it is done in a different manner. It is using the app, the features of the app. So I put up a question, there is a time slot of 30 seconds, student answer, I get the answer, how many did correctly, how many did incorrectly, and then we are able to analyze it. Here on YouTube, when we are conducting it, I'm showing you the question, you write A, B, C, D, and then we are discussing it. So it is slightly different there. Rather, I would say it is a good experience when we do a poll. This iconic subscription, which I told you in previous class also, it is a newly introduced subscription. Here you get added features like personal coach, study planner, test analysis, plus all the benefits of the Plus course, right? So this is just a comparative thing that you get what you get in Plus and what extra you get in iconic. Now this is the subscription plan, and I have shared this earlier also with you. For Plus you have all the options 1, 3, 6, 12 and 24 months. And whatever is the price, right, if you use this code you still get 10% discount on that. And this is for iconic, and here you have the option of going for 6 month, 12 or 24 month, and whatever is the pricing, again if you use this code while you take the subscription, you will be able to take 10% discount. Now another important thing is, whenever you are on Plus or iconic subscription or plan, their mock tests are conducted. And suppose I'm teaching you a particular chapter here on YouTube and you take the mock test there, and if you're able to score very good, then you know that the way you're working is helping you. And the tests, mock tests, they are prepared by experts, keeping in mind your competitive exams. So that is why the subscription is important. Now let us see what are the energy gains in various steps of aerobic respiration. Okay, Hiren says you do attend many quizzes on Unacademy and they are helpful. That's very good. Rahul is asking, ma'am, after 13 November, will you be uploading videos on both the channels or only on this channel? See, I will be giving some recorded videos and they will be uploaded maybe a little later, but I will not be conducting any live class after 13. So even if you get certain videos, those are going to be recorded videos. Okay? Alright, let us first now take up these steps and what is that energy amount or gain that we have in aerobic respiration. So this is basically a summary of what we have studied so far, and then we will come to the shuttle system. So before shuttle system, let us have a quick recap of energy. So what we'll do is we'll make a table. We'll write down the steps. We will also write down what are we getting in these steps. So here we will write the steps, the number of the steps, and we will talk about what we get. Did we get NADH2? Did we get FADH2? Did we get ATP? And then the total. ATP. We'll convert everything into ATP and then at the end we will have a grand total. So if you're talking about the first step is glycolysis. Okay, how many NADH2s do we get when we talk of glycolysis? So when we make this kind of an energy sheet, it also helps us in recap of the entire thing. So tell me, in one glycolysis, how many NADH2s are we getting? Rahul says 2. So 2 NADH2, and we know 1 NADH2 is worth 3 ATPs. So now we know because of electron transport chain and oxidative phosphorylation. So here we will ultimately get 6 ATPs. Okay? 2 molecules of NADH2s each worth 3 ATPs, so 6 ATPs. Do we get any FADH2s in glycolysis? Rahul, Amisha, Hiren, Reshmi, Reshma, they have all given the answer as 2, which is correct. Do we get any FADH2s in glycolysis? Hiren says no. What about others? No. Reshma also says no. So there is no FADH2. So if there is no molecule, no ATP get. Do we get any ATP in glycolysis? Everybody has given the answer as no for FADH2, which is good. Now tell me about ATPs. How many ATPs do we get in glycolysis? 1, 2, 3, 4. How many? Rahul says 2. What about others? Rashmi, Reshma, 2. Hiren also says 2. See, we got 4. But then we had invested 2 in the energy investment phase. So when we do that balancing out, when we return those, we are left with 2 ATPs. So in glycolysis, the total ATP gain becomes 8. Okay? Now let us come to the second step, and let us remember, we are talking about the breakdown of one complete glucose molecule. So now there will be link reaction. What do we get in link reaction? Do we get any NADH2? Do we get any NADH2 in link reaction? In one link reaction, that is pyruvic acid changing into acetyl coenzyme A, one NADH2 is obtained, and because everything is going to happen 2 times, because of 2 pyruvic acids, we will get 2 NADH2 again into 3. So here we will get 6 ATPs. Any FADH2, Hiren is writing 1 from 1 pyruvic acid. Okay, so what we have to remember is glucose giving us 2 pyruvic acid, giving us 2 acetyl coenzyme A, and there are 2 Krebs cycles because we are starting with 1 glucose. Okay, so no FADH2, no directly ATP, so here straight away we get 6 ATPs. Now let us come to the third step, that is Krebs cycle. Okay, now tell me about Krebs cycle. How many NADH2s do we get? Three NADH2, 3 into 3, 3 ATPs. So this will be 9 ATPs. Okay, but remember we have to double it. So into 2, because in one Krebs cycle we get 3 NADH2s. So into 2, so we will get 18 ATPs. Remember there are going to be 2 Krebs cycles. Okay, how many FADH2s will we get? One Krebs cycle gives us 1, one FADH2 is worth 2, so we will get 2 ATPs. But again we have to remember that we have to double it because everything is going to get doubled, so we will get 4 ATPs. Okay, how many ATPs do we get in one Krebs cycle? In one Krebs cycle we get 1, but here there will be 2 Krebs cycles, so we will get 2. So now count how many ATPs are there in total Krebs cycle. 18 plus 4. Total is going to be all. This 18 plus 4 plus 2 are to be added. How many? 24 ATPs. Now count. How many? What is the grand total? Because in electron transport chain and oxidative phosphorylation, it is just getting those NADH and FADH2 converted into that proton gradient, and by using that proton gradient, ATP synthesis. So here our grand total is going to be 38 ATPs. complete aerobic breakdown of glucose is going to give us 38 ATPs. But in most of the cases, we say that 1 glucose gives us 36 ATPs. So why this difference of 2 ATPs? Can anybody tell me why this difference of 2 ATPs? 36. Most commonly it is written as 36 because at the end you will get 36 in your hand. So total produced are 38, but what you get which can be used is 36. Now Rahul says shuttle. Okay, what exactly is this shuttle? See, we need to understand that let us take the shuttle system and we'll understand this. And there are 2 shuttle systems that we have to talk about. See, if you are talking about this mitochondria where all these reactions are taking place, and we will just draw a part of it, okay? And the outer part where the mitochondria is present is cytoplasm. So in cytoplasm, glycolysis takes place, which gives us 2 pyruvic acids. It gives us 2 NADH2 and 2 ATP. What happens to this pyruvic acid that we have understood? It takes part in the link reaction. So here is this link reaction. Link reaction is giving us 2 NADH2. Okay, so these are going to come in. Now Krebs cycle also takes place here, which is again going to give us NADH2, FADH2 and ATP. So all those checks which we are talking about, NADH2s and FADH2s, they are inside the mitochondria, except for this one. Except for this one. And the mitochondrial membrane is impermeable to NADH2. So if you have a check, rather you have 2 checks, each worth 3 ATPs. And if that check doesn't go up to the counter where you can encash it, so those 2 checks are going to be waste. So we want energy from those NADH2s which are produced as a result of glycolysis. NADH2s cannot penetrate the membrane. They cannot get into the membrane or inside the mitochondria. So what is the other option that we have? And that is where the shuttle systems work. There are 2 shuttle systems. First one is, and where are these shuttle systems? They are present in the mitochondrial membrane. And what are these shuttle systems going to do? They're going to help in encashment of this NADH2. How is it going to work? That we will say. Okay, Amisha says in eukaryotes 36, in prokaryotes, what is the number of ATP? She says in eukaryotes, from 1 glucose we get 36 ATPs. In prokaryotes, how many ATPs will we get? We get 38 ATPs. So don't you think these prokaryotes are more evolved than us? They are getting more ATPs out of 1 glucose molecule. So I guess they are better evolved. Or is there something else, some other reason? Okay, I'm writing one which is called glycerol phosphate shuttle. Now what happens is, suppose this is our mitochondrial membrane, and this glycerol phosphate shuttle, it is basically a protein glycerol phosphate thing. It is an intrinsic protein which is going to act as a shuttle. Shuttle is something which is going to help in short-term transport of things. So now this is the outer side and this is the inner mitochondrial side. So one is outer, that is cytoplasmic. This is cytoplasmic side, that is outer, and this is inner, that is mitochondrial. Now here what happens is, in this shuttle system, NADH, NADH2. Which NADH2 are we talking about? This is the one which is obtained as a result of glycolysis. It dissociates. It dissociates into NAD and a pair of protons. Now this pair of protons. So this proton goes through this shuttle system, and now this pair of proton has entered into the mitochondrial matrix. Now when this proton enters the mitochondrial matrix, it is accepted by FAD and we get FADH2. So what was produced as a result of glycolysis? NADH2. NADH2 cannot enter mitochondria. So what happens here is NADH2 dissociates and the shuttle system helps in transportation of that proton pair. So now the proton pair comes inside the mitochondria. And now it is accepted by FAD. FAD gets reduced and we get FADH2, which is worth 2 ATPs. So in glycolysis there were 2 NADH2s were formed, which we calculated as that they are worth 2 ATPs. So we will get 6 ATPs. Okay. Now when this proton goes in, 1 ATP is required. So one thing is that you are getting a molecule which is going to give you less ATPs. 2. To pump that proton, you are spending 1 ATP. You got total 38. You have spent 1. 37 left. How many NADH2s are there? 2. So how many ATPs will you spend? You will spend 2 ATPs. So 36 minus 2 ATPs to transfer these 2 NADH2s. Okay. So if you write this from glycolysis, this is from glycolysis. So here we will have to write 2 ATPs. And that is why it is 38 minus, okay, this is for prokaryotes. It is 36 ATPs, 38 minus 2 to transfer NADH2 across mitochondrial membrane. But remember, we are not going to transfer NADH2. It is going to be only proton. But we just say that we are transferring NADH2. But actually it is the proton which goes through the shuttle. So now, because you are spending these 2 ATPs here, your gain becomes 36. In prokaryotes, there is no membrane mount structure. So NADH2 which is produced as a result of glycolysis is in the cytoplasm, and rest everything is also happening in the cytoplasm. So there is no ATP required to push those NADH2 somewhere. So here basically if they undergo complete aerobic breakdown of glucose, they would get 38 ATPs. Is the first shuttle clear? And if it is clear, then we will write down the second shuttle system. Is first shuttle system clear? Okay, second one is malate aspartate shuttle system. The second shuttle system is called malate aspartate shuttle system. Again, everything is same. It is in the mitochondrial membrane. This is the membrane. This is inner side. This is outer side. And here is that shuttle. It is malate aspartate. Now again, the two NADH2s which are formed, two NADH2s which are formed as a result of glycolysis would dissociate into two NAD and two proton pairs. Okay? Now, how will these proton pairs go in? The proton pairs will go through the shuttle, and to push these two proton pairs, we would have to spend 2 ATPs. So we got 38, we have spent 2, and that is why we always say that we get 36 ATPs. Because 2 ATPs are required to push those NADH2 protons into the membrane. But here there is an added feature or an advantage. This pair of protons, two pair of protons, they are accepted by NAD, two molecules of NAD, and we get 2 NADH2. Here also is 2, so this will be 2 plus 2, 4 ATPs. And here it is going to be 2 into 3. So this will ultimately give us 6 ATP. So which shuttle system you think is more efficient? Two shuttle systems. Which one is? Why 2 are required? Why 2 ATPs are required? See, one NADH2 gives one pair of proton. Second NADH2 gives one more pair. So to push one pair, you need 1 ATP. If you have to push the second pair, you will need one more ATP. So 2 ATPs are required. Clear? So which shuttle system is more efficient? Because now what has happened is we have calculated everything. Total we got 38, and now we know we have to spend 2 here. So we will get 36. But if glycerol phosphate shuttle system is working, you don't have NADH2 here. So instead of 6, you will get 2 ATPs less. So instead of 36, that means through glycerol phosphate shuttle, one glucose gives you 34 ATPs, and through malate aspartate, one glucose will give you 36 ATPs. So it also depends on which shuttle are we using. So more efficient is malate aspartate, and whether it is malate aspartate or glycerol phosphate, you have to spend 2 ATPs to push those protons inside the mitochondria. But if it is glycerol phosphate, you don't get, see, you got the protons from NAD, and here the molecule that is synthesized is FAD. So basically you are getting 2 ATPs less. Here the proton is coming from NAD and you are again getting NAD. So you get whatever number of electrons that we have calculated. Do we use these shuttle systems randomly, or we use both? See, what happens is, the most commonly we have both the shuttle systems, and the normal shuttle system which is working is malate aspartate. But suppose if malate aspartate is overloaded, then in that case glycerol phosphate shuttle system also works, because our body also wants most efficient systems. So if everything is normal, then the malate aspartate will work, but if there is overload of this, or if there is some issue with that system, then in that case glycerol phosphate shuttle will work, or shuttle system will work. Okay, so both shuttle systems clear, and we now also know the energy balance sheet, from where we are getting how much energy. So if a question is, in eukaryotes, complete oxidation of glucose will give you how many ATPs? If that is the question, what is going to be our answer? Suppose we are talking of one glucose undergoing complete aerobic breakdown in eukaryote, then how many ATPs do you think we will get? Our answer is going to be 36. Perfect. Rahul has given the answer. The reason why we are taking 36 is because we assume that the most common shuttle system is working. Now, if the question is changed that in eukaryotes, complete oxidation of glucose where glycerol phosphate shuttle system is working, then how many ATPs will we get? Is the question clear? Complete oxidation of glucose in eukaryotes where glycerol phosphate shuttle system is working. Okay, Rahul, Amisha have given the answer 34, because in this system FADH2 will be obtained. So here there is loss of 2 ATPs. Okay, Reshma says 36. No. If glycerol phosphate is working, see, the molecule which you sent in was worth 6 ATPs. 2 NADH2s were worth 6 ATPs. After they went inside, they got converted into a molecule which is worth 4 ATPs. So there is anyways 2 ATPs less. So if glycerol phosphate shuttle is working, we get only 34 ATPs. But when nothing is mentioned, then we assume that the most common type of shuttle system is working, that is malate aspartate, and that is why we always say 36. And in prokaryotes, if the whole process takes place, the ATP gain is going to be 38, and the reason is there is no membrane inside. So no energy required to pump that proton from NADH to anywhere. So everything is in that cytoplasm. Amisha says, how do we know kaun sa wala kab kaam kar raha hai? You're talking about the shuttle system. I said most of the times it is this working, malate aspartate. But if malate aspartate is overloaded, then only it is going to be glycerol phosphate. So shuttle systems clear? Any confusion in shuttle systems? So we have done two things so far. One is the energy balance sheet. How do we get this 36 or 38 ATPs? And then the shuttle systems. Because our balance sheet says 38 ATPs and in hand we get 36 ATPs. So this is a question that where do these two go? What can be the factors कि वो overload हो जाए? See, if there are more and more NADH2s, more and more NADH2s produced, so this will be overloaded because through the shuttle it takes time for the protons to pass. So if there is accumulation of NADH2 in the outer part, that is in the cytoplasm, then the other system will also help. It will give us little less ATP, but at least the protons will start moving in, and when they go in, the other things start, that is electron transport chain, oxidant, because ultimately you want ATPs, right? So if there is an overload, then the other system will work. Is it clear now? Amisha, the question you asked, is it clear now? That's very good. Alright, so these two parts we have done. Now let us talk about the efficiency of aerobic respiration. For this, we'll have to compare aerobic and anaerobic respiration. Efficiency means how efficiently a particular system or type of respiration is working. Ma'am, Rahul is asking, ma'am, is there something in which ATP is in excess condition? See, our body system or cells, they work in such a manner that energy is produced only when it is required and in the quantity in which it is required. So the process goes on in that manner only. ATP can be stored in the cell, so if there is little extra, it can be stored also, but not too much. Okay, now if we are talking about efficiency, that means if one glucose is broken down aerobically, how efficient it is means how many ATPs, and if one glucose is broken down anaerobically, then how efficient it is, how many ATPs are obtained. So one glucose broken down anaerobically gives us only 2 ATPs, and similarly one glucose broken down aerobically gives us 36 ATPs. So don't you think aerobic respiration is about 18 times more efficient? Aerobic respiration is about 18 times more efficient because the substrate is the same, it is the glucose. So if glucose is broken down in absence of oxygen, you are getting only 2 ATPs, and when it is broken down in presence of oxygen, then the process is more efficient because you are getting 18 times more ATPs. Okay, now I am going to give you one more term here. It is called protoplasmic respiration. What is meant by protoplasmic respiration? Have you heard this term before? What is this protoplasmic respiration? Have you heard this before? Normally, what is our respiratory substrate? Respiratory substrate is glucose, or we write carbohydrates. Okay? If the respiratory substrate becomes protein, or in other words, if we say that instead of carbohydrates, if the proteins are broken down, then that respiration is called protoplasmic. If instead of carbohydrates, proteins are broken down to release energy. Then that type of respiration is called protoplasmic respiration. Okay, let us talk about one more term. And you know, when we normally break down the reserves, the sequence is carbohydrate first. So whenever energy requirement is there, it is carbohydrate which will be broken down. If carbohydrate is finished, then fats are broken down. And when fats are finished, then proteins are broken down. So proteins are the last ones to be broken down. And that is why people who want to lose weight and are doing dieting and they eat less, thinking that in 2, 3, 4 days there will be weight loss. So initially when you stop or reduce your calorie intake, then all that glycogen which is there in your muscles and your liver is broken down. When that glycogen is gone, and when glycogen is gone from the muscle, there is no loss of the weight, because your weight is because of fat. And in 2, 3, 4 days or 8 days of our dieting, we see that we have stopped eating our favorite food and the weight is as it is, because we step on the weighing scale every day, right? So we find that there's no weight loss, and then we get demotivated and we start eating everything. So unless and until this carbohydrate, the glycogen is completely finished, fat will not break down. So if you want to lose weight, you will have to continue it for a longer period of time. And when fat is finished, then there is protein. And if protein breakdown starts, then that is dangerous. If it is carbohydrate, if it is fat, it's okay. Because when proteins break down, there are ketone bodies which are formed in our body. And then it starts affecting our vital organs like brain and heart and everything. Okay, that's why you should study biology. Hiren, I like that sentence. Yes. So, you know, when people go for a diet or they go for this dieting, they don't realize. And that's why I say biology is everywhere. So, all around us, what we do, it's all biology. And that's why we need to study. See, if you study it this way, this particular thing, now, do you think you'll have to mug it up? That what is broken down first, then what happens, then what happens? Now you have understood it, it's going to stay with you forever. Okay, this part clear? Now let us add one more term, that is amphibolic pathway. Now what happens in respiration? Amphibolic word is used for both a process in which there is anabolism and catabolism. Amphibolic process is a process which has anabolic and catabolic pathways or steps. What are anabolic steps? Where something is being built up. Catabolic, when something is broken down. So now in case of respiration, anabolism is also taking place and catabolism is also taking place, and that is why this respiratory pathway, that is the complete aerobic respiration pathway, is called amphibolic pathway. Okay, so protoplasmic respiration, amphibolic pathway, these are two important terms which we have studied. Now in this part, is there anything which is difficult? The part which we did earlier, and then we will come to the recap. See, we did shuttle system, we did the energy gain. From various steps, how much energy are we getting? And if we write 36, if we write 38, what exactly it means? So any confusion in this particular energy sheet? No doubt, that's very good. What about others? Okay, if there is no doubt in this, then let us come to the shuttle system. What is the shuttle system for? The shuttle system is to push those two protons which are released from NADH2 of glycolysis into the mitochondria so that they can be used to generate ATP. Now these protons, they cannot pass easily. They have to be pumped in actively, and whenever we use the word pumping in, active, ATP has to be used. So in in glycolysis, two NADH2s were produced. So the proton of one NADH2, when pumped in, 1 ATP, protons of second NADH2, second ATP. And that is why, though we get 38 according to that sheet, we ultimately get only 36 because we have to use 2 ATPs for this. Shuttle system is the protein or the molecule which is passing these protons. If it is glycerol phosphate, we get 2 ATPs less because the protons are taken from NADH2 and accepted by FAD. In the other one, that is malate aspartate shuttle system, the protons are given by NADH2, accepted by NAD. So in the second one, the energy gain doesn't change. So here 36 ATPs only. So this one is more efficient as compared to the glycerol phosphate. Okay, protoplasmic respiration, amphibolic pathways, these are two more terms that we have talked about. All right, now coming to the recap. Now tell me anything that is difficult in these two processes which we have studied. That is aerobic respiration and anaerobic respiration. In anaerobic respiration, we said it is known as fermentation. You can ask the doubt when I am going over it, okay? You can just send your question. Herein is NADH2 versus NADPH. See, there is a difference of phosphate here. That is the only difference. Otherwise, it is nicotinamide adenine dinucleotide. It is nicotinamide adenine dinucleotide phosphate. And this works in plants or found in plants, and this is found in both plants and animals. This is associated with photosynthesis, and this is associated with respiration. How to memorize complexes? Which complex are we talking about? The electron transport chain? Ayush? Are we talking about electron transport chain complexes? Yes, okay. I told you to draw that electron transport chain once. Did you try drawing it? Did you see the diagram which is given in NCERT, and did you try to draw it? If you draw it once or twice, you'll remember. See, I can tell you when we saw that diagram, we said that electron which is going through complex number 1. The complex number 1 was having FMN and FES. Are you able to recall? Then there was complex number 3, which had all different types of cytochromes. Cytochrome B, cytochrome C, and all those. Then there was complex number 2, which was an enzyme. This is the only enzyme in Krebs cycle, which is in the membrane. Krebs cycle takes place in the matrix of mitochondria. but the enzyme succinate dehydrogenase is in the membrane. That is complex number 2. And then there was complex number 4, which was again having cytochromes. So if you draw it once, you write those things down, and try to see that image of electron transport chain, which I showed you yesterday, plus it is given in the NCERT 2-3 times, so that visual memory will also remain with you. So when you close your eyes, okay, first was FMN, then was FES, then there was the third complex which had cytochromes, then there was this different shaped enzyme. You remember the shape of the enzyme? It was something like that. This you get when you draw it. So see it and draw it. You will remember those things. It is not that difficult. But for that, you will have to draw it at least 2-3 times. Okay? Alright. Anything else? So we were talking about anaerobic respiration, which is known as fermentation. 2 types. One is lactic acid fermentation, other is alcohol fermentation. We used the term homo fermentation and hetero fermentation. Homo is when you get only one product, and hetero is when you get more than one product. So lactic acid fermentation is homo because you get only lactic acid. Alcohol fermentation is hetero because here we get 2 things, ethyl alcohol and carbon dioxide. Ask questions of this chapter. Yes, this is what I have planned for tomorrow. We will be having a quiz tomorrow. So tomorrow's class, we have to just talk about one small thing and then it is all quiz. And that is why I am asking you the doubts. If you have doubts, we can clarify those doubts here. And tomorrow, after discussing the last thing, that is a common pathway, we will straight away start with quiz. So we'll get maximum time for question solving. Okay, what about respirometer? What about it? You've written respirometer. It's a device. So what about this device? Oh, structure. We don't have to do the structure. It's a machine. We don't have to do the structure of respirometer. Any other question related to aerobic and anaerobic respiration? See, our syllabus anyways is very vast. So we should not be going into things which we don't need right now. Aerobic, anaerobic respiration, all steps, energy gain, Shuttle systems, efficiency of which respiration is more, all these things are done. Now, tomorrow we will be talking only about the common pathway. That is a common pathway for carbohydrate, proteins and fats. Because we know fat is also broken down, protein is also broken down. So how do they give us energy? Is there a common pathway? That we will talk about in the tomorrow's class. Okay, so I was talking about there is some change, and that is one lecture, not a lecture, I have planned a small video for you. It is going to be a live video, and that is on Saturday at 2 o'clock. And that will be focusing only on the strategy part. So those students who join little late, this is for them, that there will be a video or a lecture or a session on a channel, YouTube channel. It's called Biology 101 at 2 o'clock on Saturday. And the main focus is going to be the strategies to score 360 in biology. This is what we will be talking about. And then in the month of November, I won't be taking classes on other YouTube channels. I'll be taking classes only on 2 platforms. One is YouTube channel, this one, that is Let's Crack NEET UG English, and the other is going to be on Unacademy Learning app, which is a special class. And for your information, I'm starting the second chapter of genetics. We have a poll tomorrow, and after that we will be starting with the second chapter, that is molecular basis of inheritance. So if you want, you can join in there also. Amisha, timing of the special class. See, what I'm planning is, I'll share the schedule with you, but I'll give you some idea that I'm planning to have special class from 11 to 12, and then 12:30 to 1:30 your class. That is on this channel. Okay, but when we meet tomorrow, I'm working on the schedule, and as soon as the schedule is with me, I'll share it with you so that you know which class is conducted where. So just for your information, second chapter of genetics, molecular basis of inheritance, that is what I'm starting on the special classes, but that is after I think from 31st or maybe Monday. Okay, Amisha, yes, I'm in 11th. You know, when we do this molecular basis, the first part is of 11th only, when we have to talk about the structure of DNA and RNA, which is like the main molecules. So that is going to be common for 11th and 12th. So there's no harm if you attend that class, but for that you will have to install the app. So don't forget to do that. All right, so tomorrow I'll see you at the same time. And we will be having, this we have done, and we will be having a quiz after we finish that small part which is remaining. And don't forget to subscribe, take the subscription and use this code so that you get the discount. So till tomorrow, keep practicing the chapter. We will have a quiz tomorrow, and I hope you all do very, very, very well. Is NCERT sufficient for 360? Yes, we are doing everything from NCERT only. But you need to understand that in NCERT there are some hidden things also. So that is the hidden thing that we need to understand. Okay, so bye for now, and we will meet again tomorrow with the quiz. Bye everyone.